Welcome to this presentation on wild and natural brewing given to the Institute of Brewing Great Northern Section meeting on the 7th of March 2019 in Sunderland at Brewlab. Now, the title of this wild and natural brewing focuses really on the microorganisms involved in brewing, with perhaps a particular application to novel beers, beers brewed with different organisms, mixtures and so on. And to begin with, let's have a quick overview of the range of organisms which we are acquainted with really as microorganisms. And these cover everything from viruses through to protozoa. And quite quickly we can rule out viruses and algae and protozoa as contributors to any of our brewing operations, at least at the moment. Really we want to look at the true bacteria and the fungi, yeasts particularly, molds possibly, as being organisms which could become involved. Now we've got some examples here. We can see bacteria, molds and yeast in their standard characteristics that we would see in the microscope or on agar plates. Now recent work has compiled together the range of bacteria, so let's look at that as a major group. And if we pulled most of the bacteria groups together into a sort of tree of life almost, we'll be able to say that there's quite a lot. There's a big range of microorganisms into the bacteria group. So if we were to look in the environment, we would find some from many of these groups in any natural sample. For the ones that would be relevant to ourselves, which would produce perhaps an interesting worked fermentation, ones that would produce a safe worked fermentation, we'd perhaps focus on the groups here, Firmicules particularly, maybe some of the affiliated ones near that group. Now, in a way, there's so many organisms here, many of which, to be honest, haven't perhaps even been observed in fermentation, certainly. So there's plenty of opportunity to develop investigations on these lines. A little bit more of a focus on the more relevant groups gives this sort of grouping here, where we've got the Firmicules and the proteobacteria as sort of adjacent larger groupings. And we can recognise some of these, the Lactococcus, the Streptococcus, the Staphylococcus, and then the Bacillus, the Lactobacillus group, as being prevalent in this. Oniococcus, Bacillus, and so on, would be ones that might be worth considering. We've got others in the same sort of group, Clostridium, we don't want to be involved with, and we've got others in the Proteobacter group that, again, would stay well away from the Escherichia. But then again, there are some natural Escherichia, or maybe that sort of group, which would naturally occur and ferment in worts, wouldn't really resist the alcohol or the acid. And we want to stay well away from Campylobacter and Chlamydia, these sort of things. So we've got to be fairly careful in what we allow to grow. But thankfully, the work we use is relatively protected with the acidity, the alcohol, the hops and so on. Now if we looked further down into the breakdown of the bacilli groups, we would have the Lactobacilliales, we'd have the Lactobacillus grouping here, we'd have Pediococcus, which we're fairly well acquainted with, not that we'd want these in our standard beers by any means. Leuconostox, Oniococcus present in cider and wine. Then there's Lactococcus, Streptococcus. Many of these would naturally be able to grow in wort to some degree. There's others which we would probably be aware of. Bifidobacteria are well known as probiotics. So there's a sort of interesting range that we could consider as being relevant to some of our potential wild brewing. Now if we just went out into the environment and isolated organisms, what would we get? Well, here's an agar plate with an air impact study, and we can see lots of colonies of mould. Now these black moulds, who knows what they are exactly, but could well be penicillin or aspergillus, and some of those produce pretty unpleasant toxins and so on. So we've got to be careful as having those in our works, at least at the beginning, even if they don't continue and do a fermentation. But there's also bacteria in there, and we don't know what those are, but those yellow ones might well be Staphylococci. So we get a whole range, and to be honest, moulds are very common in most airs, in most buildings. If we had selective media that would sort of exclude the moulds to a degree, we'd get more bacteria and maybe yeast. So some of these might be yeasts, the green ones, but some of the red ones maybe, again. So there's a range of ones that we can isolate, but we need to do further study to be able to be sure that they are, I suppose, not hazardous. And just to look at the requirement for some competence in microbiology, here's a streak plate which is isolated a pure culture of yeast. Here's a streak plate which has been unsuccessful because the moulds have been in the air and the microbiology hasn't been so rigorous. So we've got to be careful we don't get contaminated cultures coming through. Now we could use mixed or pure cultures of bacteria. Here's a mixed culture with yeast and in fact a crystal of probably oxalic acid. You can see long rods which probably be lactobacillus and so on, and in this one here we've got sort of short and very long rods, so a variety of things can appear and we can purify and then identify. And to identify we'd be using some probably either classical techniques looking at their physiology, but nowadays it's much more looking at the DNA isolation and then matching against. We'd be looking at databases that would be able to do a sequence of the DNA bases, such as this one here, to be able to match it. There are other methods, MALDI-TOF, which are based on protein characteristics, but for most brewers it's going to be most likely to be more rigorous to look at the DNA profile. Now when we go into the yeast groups, what we find now is that we've got two big groups relevant with yeast, Ascomycetes and Basidiomycetes. There's other groups of fungi, the chytrids and the Zooomyces, but on the whole any of the yeasts that we're going to be have potential for brewing with would be in these two groups. The Saccharomyces of course are here, but there's plenty of others which will naturally occur and which will be isolated from many environmental sources. Now looking at the sort of evolution of yeast, it's interesting to go back in time and go back 1 billion years or more and be able to say, well, there's fungi appearing 1.5 billion years ago, breaking it into the Basidiomycetes and the Ascomycetes. And the Ascomycetes produces 3 groups, and one of those 3 groups, the Saccharomycotinia, produces the Saccharomycetes and affiliated genera, Candida, Debromyces, and so on. So these are probably the most likely ones, and seems to have been found to be ones which can grow in high sugar concentrations. Now it's interesting to look at the affiliated characteristics, that over the same sort of period of time of the evolution of these yeasts, mushrooms and smuts and rusts come from the Basidiomycetes, but we also have the flowering plants evolving, and with Saccharomyces and these other yeasts being naturally growing in flowers, in the nectar, in fruits and in bark of trees and so on tree sap and such, that they would have naturally colonised those environments, and that's why they have developed in this sort of period of time. Now people recently have done studies on the whole genome sequence of a range of yeasts. So here's 100 or so yeasts which have been all fully sequenced and compared, and it's shown clusters have developed into areas. So beer 2 and beer 1 show 2 big clusters of beer yeasts. But there are other yeasts of wine and ones from different parts of the world which also cluster separately. And it's interesting that the yeast from Belgium, Germany cluster separate from the US and from Britain. Beer 2 does seem to have other characteristics, and some groups have then done some further genetics and determined what genes are present in these groups. So it's interesting that the same sort of grouping here is present in beer and sake and wine and so on, being separate, but in the beer group we've got wheat beer, English Irish ales and German beers, and it's shown that certain genes such as the phenolic off flavour genes and the maltotriose uptake genes are active, with this signature here that the black dots show activity in the genes, are active in wheat beer yeasts but not so in our standard brewing yeasts, which is why we don't have the POF characteristic and we don't have full maltotriose uptake necessarily in all of these, in our standard really, brewing yeasts. So it's interesting that now we can start to characterise yeast for their particular genes and be able to say what those impacts will be in the brewing. And here we are with more details, closer up details of those, and we can see the different clustering in the genetics. The physiology shows that ethanol production in those different groups, ethanol production is moderate in the standard beer yeasts, a bit higher in the beer 2 yeasts, and in the other yeast wine and so on would expect them to be able to produce more alcohol in the fermentation. And the maltotriose uptake is good in beer yeast but very poor in wine yeasts, again as we might expect. Now, to isolate yeasts and so on, what we need to look at is both the morphology, the appearance, the physiology, their ability and their genetics. But we'd start off with the raw material from the environment. We'd then maybe plate it out on an agar plate. We'd isolate and look at the individual cells from those colonies. And here's those individual yeasts, or some of them that we isolated in this particular case from that fruit. So we found 2 Saccharomyces cerevisiae were present, and 3 other different species. Now here's the surface of the fruit, and it shows that there are clusters of organisms present as colonies. Most of them are filamentous, but there will be yeasts in there. And when we looked at the physiology of these yeasts, we found that the 2 Saccharomyces, one produced alcohol at a reasonable level compared to a control yeast from a 1040 wort. We got 4% with the control yeast, 3.5% with the Saccharomyces number 1. The other yeasts didn't produce very much alcohol, but they did have different flocculation characteristics. So it's valid to say that really there's a community of yeasts there which probably work well together, but we need to maybe isolate and selectively culture some of them, particularly that one. Now when we compare yeasts for their ability, their physiology, we get different responses. So here's 8 yeasts grouped with 2 ales, 2 lagers, 2 wheat beers and 2 ciders, and growing under different conditions. 10 degree Plato wort, 1040, 20 degree Plato wort, 1080, after an acid treatment and after a exposure to hydrogen peroxide. Now these are the sort of stresses which yeast would be under in their fermentation conditions. So growing in the 1040 wort, yep, standard profile, we see that that's fairly consistent, although some yeasts don't produce as much growth as others. So again, they're going to vary to some degree. In the very strong high gravity wort, we get a slowing of the rate of growth, and we also get a delay in the time at which the growth starts. That's also prevalent in the acid wash condition, but all of them do recover. But under the oxygen conditions, we do get at least the ale yeast and the lager yeast just don't work at all. One of the wheat beer yeast did, but 2 of the ciders did. But they are very delayed in their growth, indicating the severity of oxygen excess to many yeasts. Now we did further studies in this particular case with a mixture of yeast, a MegaBlend, where in fact a mixture of wild isolation. So people came together and put a mixture of their isolates together and then further went and brewed with it. And we did a next generation sequencing on this to identify the range of organisms present. And as we can see, there's a lot of acetobacters and a lot of lactobacilluses, a couple of pediococcus. And it's interesting that the sequence of the growth of these differs. So in the 2 Mega 1, Mega 2, Mega 3 show the periods during the fermentation: early fermentation, mid fermentation, late. The acetics are common at the beginning, where probably there would be levels of oxygen, less so in the middle, but prevalent at the end. The lactics are prevalent in the middle and the end, which again we might expect. With the yeast profiles, we found the Brettanomyces were very prevalent in these. Saccharomyces less so, but really only growing towards the end of the fermentation. So if we are looking to use these wild yeasts, we're going to get the characteristics from Brettanomyces most dominantly. Now we can use molecular biology to identify particular genes. So where we've looked at the physiology, we can go into the genetics and say, is there a particular gene there? So the HORA gene is common in Lactobacillus, giving hot tolerance. And we find that in the megablends, that all 3 megablends had that gene in it, and the control Lactobacillus shows the presence of those genes. So these are the positive results against a negative control. Another one that's particularly relevant is the diastaticus. Now, diastaticus, as doubtless aware, is the problem where we get overcarbonation in a beer and gushing. So really don't want to have a beer which is overcarbonated and going to produce a forced exit of gas. This is a commercial problem because we don't want to be releasing commercial beer into the market, which could be hazardous. Now, another identification that's quite important is to check that we haven't got toxic or hazardous organisms. And some of these we want to avoid definitely is food spoilage organisms such as E. coli, Salmonella, Campylobacter and so on. So again, we could use the same genetics to test whether they're there or not. So these are the controls for Salmonella and E. coli, and these are the negative beer samples. So that would give us really an assurance that the product was safe. And there's always a potential, if we're selling beer into the community, that there might have been a contamination according to where our environmental sampling was taken. Of course, it could be poor aseptic techniques. We're just going to make sure that we don't have those organisms ourselves. Here's another study that we did with just looking also at identifying organisms present in this case spent grain to see whether they would be of use either in fermentation of beer or products or maybe of biotechnology of use. So we did find in the microorganisms at different times of incubation of spent grain particular organisms which were prevalent and may well be digestible or allow digestion of some of the spent grain material, and the same with the yeasts that were present there. So there's work that can be done and is being done in many areas to look at what organisms are relevant to our brewing operations, different than our standard ones that we've been using for the last 2 centuries. Another example is to have a bit of a screening as to what's on the market. So here's a quick profile of 6 different sour beers, assessed for their ABV, their gravities, bitterness, colour and their profile of acids. So what we find here is that the alcohol is pretty low, except for one sample. The gravity is relatively low as well. The finishing gravity is mostly low, except one which, although it started at 1043, finished at 1013, so that had a reasonable mouthfeel, but 2 or 3 of them are very low. Bitterness also very low, and colour very low. So there's not a lot of character in these beers, as we would expect in a standard beer. However, the acidity is prevalent, not as acetic very much, but certainly lactic acid. Citric acid a little bit present, but really it's the lactic acid which is the dominant acid present, and will give the certain character, and probably a good character rather than the sharpness of acetics. Now, looking at the microorganisms present, it is apparent that there are microorganisms in all of these 6 samples. There's yeast in 5 of them, and there's bacteria only in 1, and there's yeast only in another. But we can see the bacteria certainly in number 6 is long or medium rods and long rods, short rods perhaps in number 3, a mixture in number 2, a mixture in number 5, and in number 1. So obviously these beers are produced with appropriate microorganisms. Now, what's our options? What can we brew with? Pure yeast, yes, of course. We've got plenty of isolates, we've got plenty of culture collections, which can give us different yeast of different characteristics. We could mix them, we could have 2 or more strains together, which would have been traditional even only 50-60 years ago. We could use bacteria, we can isolate particular bacteria, we've seen a number so far, and we could choose those and allow them to ferment. Or we could have a mixture, we could have a community, we could have a succession. This is a succession that we might find in cider. Hansenia spora develops first. Once the alcohol level rises, the Saccharomyces develops and ferments fully, and once that's finished, the Brettanomyces takes over. So we're likely to get a range of characteristics from those organisms that are present in that sequence. We could have mixed microbes, yeast and bacteria, of course, as we've just seen in those sour beers. And it's interesting looking at what was prevalent in history. So going back 100 years, some of the illustrations of the yeasts and ferments that they used do show particular yeasts, and they do differ according to their origin. Burton yeast, for example, London yeast. But it does show that the yeast was contaminated with long rods in this particular case, but in the forcing samples on this one, many of the samples do have microorganisms in. So it's likely that people years ago, traditional classic beers, were brewed with a mixture of bacteria and yeast. And again, there would be a community and a succession progressing through that. Now, here's one bacteria that we don't want, which we certainly don't want to use, because that would produce some pretty awful flavours. And what's prevalent is that it's, what's evident is that it's a motile organism. And as we can see here, that the movement of the organisms sort of gives it away, that some of them are very fast moving throughout the system. And if we get these organisms penetrating into our plants, it may well be that it's very hard to get them out. So what we want to be sure of is that we've got a very good hygiene and awareness and a sort of quality control assurance of the system. Now moving on to really the future and finishing off with what can be done. Here's some examples from the literature in the last few years as to what people have been doing with novel yeasts and microorganisms. Isolation and characterization of ethanol producing yeast, so people just screening and seeing what's available. Magnetic modification of yeast cells. Fast methods for identifying hybrids, so obviously looking to identify and characterize more easily. Mating of organisms together to Saccharomyces cerevisiae to enhance a particular flavour. Characterising Brettanomyces for bioflavourings, perhaps not just in beers but in other food products. Gene expression in wheat beer yeasts, and the use of particular mutagenesis to create brewing yeast with particular flavour changes. So there will become the greater interest in genetic modified or modified organisms which have been particularly selected for particular purposes, and those will become prevalent increasingly in a sense in the industry. It's a matter of choice for the brewer as to whether they would use them or not. So here are the studies cited in this work, which are available obviously in the academic community from journals and so on. And here's some suggestions for the future. That will be a wider awareness of brewing microbiology generally. We'll be looking to have our brewers or members present in our breweries who will understand microbiology more, to use a wider range of yeast strains and species, to have more knowledge of yeast genetics and the identification and control of genes, greater knowledge of dual and multiple strains, and potentially genetically modified or engineered yeasts that will become available. So overall it's going to be a time of change. There's plenty of people doing lots of developments nowadays. I'm sure we will see these appear in the industry before very long, and we will be looking to incorporate them, to provide our own view on those and our own developments as appropriate. So thank you for listening, and I hope it's been of interest to your microbiology knowledge.